Hello, so this video is on the required practical enzymes for key stage 4. So first of all, what is an enzyme? An enzyme is a biological catalyst that speeds up the rate of reaction without being used up. Enzymes are made of proteins. Okay, so enzyme activity is described using the lock and key model. So first of all, enzymes are specific, okay? So they can only work with specific substrates, okay? So if this substrate tried to work with this enzyme, it would not work, okay? Similar for these. So these will only fit for this one for this one, and this one for this one, okay? So enzymes are specific. Secondly, the substrates bind to the active site. Okay, so this is the substrate, this is the enzyme, and this is the substrate binding to the enzyme. Okay, so watch that again. So substrates bind to the active site. Okay, an enzyme substrate complex is formed. Okay, so when the substrate binds to the enzyme, binds to the active site, an enzyme substrate complex is formed. So the substrate and enzyme have complementary shapes. Okay, so that means this substrate will fit perfectly into this enzyme's active site. Okay, so if this active site or if this substrate changes shape slightly, it will no longer fit and cannot form the enzyme substrate complex. Okay, so when the substrate has formed the enzyme substrate complex, bonds are made or broken. Okay, so here this is one substrate. Okay, this could bind with the enzyme. Okay, and the bonds could be broken, and this will now split into two parts, two substrates. Okay, or it could be two substrates joining in the enzyme's active site, and then the bonds could be formed, and then it could release as one substrate. So, and then finally, the products are released. Okay, so again, substrate binds with the active site, enzyme substrate complex is formed, products are released. So that was the basics of enzymes. Okay, so what I'm going to show you now is the required practical. So first of all, our equipment. Okay, we've got lots of buffers at different pHs. Okay, we've got pH 2, pH 3, pH 5, pH 7. Okay, got a stopwatch, got lots of spotting tiles. Okay, we've got iodine solution, and then we have, which you may not be able to see very well, we have a water bath. Okay, at being kept at 35 degrees C. Okay, and it has starch solution, and amylase solution. Okay, both of these are at 1% concentration. So firstly, what we are going to do is we're going to take 1 milliliter. Okay, so we're going to take our syringe, we're going to take 1 milliliter of our first buffer. We're going to use pH 7 first. So let's move these out of the way. So start with pH 7. We're going to take 1 milliliter of this. Okay, so these syringes are very stiff. 1 milliliter. Okay, we're going to add that to our starch solution. It's being kept at 35 degrees in the water bath. Okay, so that is added into there. Right, so next what we need to do, we need to put a drop of iodine solution, okay, into each one of these pits on the spotted tile. Okay, it's important to only use one drop, okay, because otherwise it can affect the results. Very carefully, one drop. Okay, next what we need to do is we need to take our 1% amylase solution, okay, and we need to pour that in with the starch and buffer. Okay, as soon as we've done that, we need to put one drop. Okay, so I'm going to mix these, I'm going to put them back in for now, just to stop them from getting the temperature changing. So once we've done that, what I'm going to do is using the pipette, I'm going to scoop a load up and drop one in, one in, one in, one in, okay, all the way along this spotting tile, but at an interval of 30 seconds. Okay, so every 30 seconds, I'm going to put another drop in. Okay, so I've finished that, okay. This is what my pH 7 spotting tile looks like, okay. What you would do is you would repeat that for pH 5, pH 3, and pH. So here are the pictures of the 4 experiments that I completed. Okay, so here's the pH 7, which we started to do together, pH 5, pH 3, and pH 2. Okay, so you might be wondering, well, what was the point of that experiment? Basically, what we were trying to do is see the effect of pH on the rate of reaction for the enzymes. Okay, so in that case, the enzymes, amylase, okay, was breaking down the starch. Okay, when the solution is black like this, okay, it means that there's still starch present. Okay, when the iodine turns yellow like this, okay, it means there's no starch present.